Hello everyone, welcome to today's Science of Childhood Cancer lecture. I'm Charles Mulligan, Deputy Director of the St Jude Comprehensive Cancer Centre, and will host today's lecture. Thank you all for joining and participating in this exciting lecture series. Before introducing today's speaker, I'd like to give a shout out to next week's speaker, our own Melissa Hudson, Director of the Cancer Survivorship Division at St Jude, who will present a lecture on the role of survivorship research in advancing childhood cancer care and quality of survival. It'll be another wonderful talk, so please join for that. So with that, it's my great pleasure to welcome and introduce Olivier Delattre as today's speaker in the Science of Childhood Cancer. Dr. Delattre is Research Director of INSERM in the Department of Cancer, Heterogeneity, Instability and Plasticity. He's also the Director of the Pediatric Oncology Centre, SAREDO, of the Institute Curie, which is entirely dedicated to cancer in patients under the age of 25. By background, Olivier received his MD from Paris-Six University and his PhD in molecular biology from Paris-Sète University. Since that time, his career has been at INSERM, where he's held directorships since 1994. His research contributions in the field of pediatric cancer have been diverse, transformative and impactful, and span multiple tumour types, including Ewing sarcoma, non-Ewing sarcoma, neuroblastoma, rhabdomyosarcoma and rhabdoid tumours. And notably and extremely impressively, these studies include the identification of many of the sentinel genetic mutations and translocations that define many of the key subtypes of these pediatric cancers that remain many of the most problematic tumours that we all work on today. And his work has furthered this with very elegant dissections of the role of these alterations in driving malignant transformation, and I believe he's going to speak on one of these themes today. He's been extremely influential, and his work has inspired many of the leading researchers in the field of pediatric cancer today, so it's truly our privilege to have you join. He's published over 400 manuscripts and received many prestigious research awards during his career, including those from the French Academies of Science, National Academy of Medicine, the Charles Oberlin Great Award, and Leopold Griffel Award of the French Foundation for Cancer Research. So again, Olivier, we're all super excited to have you join today. Thank you very much for speaking, and over to you.
Okay, thank you very much for the invitation to speak today at this fantastic series of seminars, and thank you very much, Charles, for your very kind introductory works. So just 40 years ago, two cytogeneticists, Alain Orias and Claude Turcarel, described this 1122 translocation in Ewing sarcoma. And this was one of the first translocation that was identified in solid tumors. And this was the topic of my PhD thesis at this time, to identify the genes involved in the translocation breakpoint. And with my colleagues, Jessica and others, we undertook a journey over the genome to find out the breakpoint. And at this time, the human genome was not known, and there were only a very few probes on chromosome 22. And just to make a long story short, we used a series of tools that seems completely outdated now, like bacterial like artificial chromosome, yeast artificial chromosome walking, Southern blot, and so on. And after a long journey, quite interesting, it was like exploring new territories. And finding new genes in the journey, we ultimately find out the breakpoint and showing that finding the DNA fragment that was rearranged in tumors. Altogether, it was a kind of 5 years endeavors with five people. And it's quite amazing to see that nowadays, in a few hours, you can get the same results, and this witness the progress of technology and science. But I was quite naive at this time, and I thought that once we will identify the genes at this translocation breakpoint, we will find out what is the biology of the tumors and potentially find out treatment. targeting this translocation. And it has to be acknowledged that 30 years, almost 30 years after the characterization of this translocation, we don't know how to target EWS FLI, which is a fusion genes, even though this is a fantastic target because many labs have shown that if you knock out EWS FLI, you just completely kill the cells. So I hope in the next few years, and there are a lot of labs working on this, we will find the solution to target EWS FLI. So Ewing sarcoma is mostly a bone tumor, which is mostly observed in teenager, and as many childhood cancer and many cancer in general, the prognosis is highly dependent upon the presence of metastasis. And genetically, it's a very genetically simple tumor with mostly this translocation, which is almost observed in 90% of cases. And this leads to this EWS FLI fusion gene, which encode a chimeric protein. The EWS fusion protein, so the EWS gene, which is also called EWS-R1, I will come to this a bit later, is an RNA binding protein, the function of which is not clearly understood yet. Its C-terminal portion has an RNA recognition motif and can bind to RNA, and its N-terminal portion, it's a proline-like domain which is a low complexity domain, which is capable of self-association and phase transition. FLI1 is a transcription factor with a DNA binding domain and regulatory sequences on the N-terminal part. And as a result of the fusion, the low complexity domain of EWS FLI is fused to the DNA binding domain of FLI1. So many labs have attempted and succeeded somehow to elucidate the mechanism of action of EWS FLI. EWS FLI can bind to two types of motif, either the classical ETS binding motif, because FLI1 is an ETS family member, either a much more specific sequence, which is a microsatellite sequence with GGAA repeats. And the more GGAA you have, the more EWS FLI can bind to them until a kind of maximum between 15 and 20 repeats. And none of the wild-type ETS member can bind to this microsatellite sequence. And through binding to those two types of sequences, EWS FLI dysregulates a number of metabolisms and pathway in the cells. The precise mechanisms of action have also been investigated a lot. And thanks to the work of Bob Tijan's lab recently, and also Miguel Rivera with Sigal Kadosh, we begin to understand a bit what's going on the microsatellite sequences. So those microsatellites are usually completely in a heterochromatin conformation and accessible to transcription factor. And upon the translocation, next, the EWS FLI protein is produced, can bind to these sequences next. And upon binding of these sequences, what we think is that there is an increased local concentration of EWS FLI, so an increased concentration of the low complexity domain of EWS FLI, leading to some phase transition and condensate formation that were recently described at your seminars by Bob Young and his colleague. And with this, next, there is a recruitment of the transcription machinery, chromatin remodeling machinery, histone acetylation machinery, and which lead next to a completely new enhancer at this level, which can activate genes in the neighborhood. So EWS FLI is clearly, Ewing sarcoma is clearly kind of epigenetic disease where you have this transcription factor that activates sequences throughout the genome. In terms of intertumor heterogeneity of Ewing sarcoma, so there are different types of fusion depending upon the position of the breakpoint between EWS and FLI1. And initially, EWS1 was the region on chromosome 22 where translocation breakpoint occur. And now, for any reason I don't really know, this is the name of the gene. So throughout my talk, I will speak sometimes of EWS and sometimes of EWS1. And depending on the position of the breakpoint, you have different fusions. There do not seem to have any clinical consequences of this fusion, apart from the mechanisms that lead to the fusion, which can be either a balanced translocation or, as shown recently by the group of Adam Schlien, it can be some chromoplexy mechanisms. And it seems that the translocations that arise through chromoplexy are of bad prognosis as compared to balanced translocation. FLI1 is observed is a fusion partner in 85% of cases. The second one is ERG, which is very similar to FLI1, and the other one are less frequent. And between FLI1 and ERG, there do not seem to have any real differences in terms of diagnosis and prognosis and response to treatment. So Charlie Roberts recently presented rhabdoid tumors and said that they have very low background of mutation, and this is approximately the same for Ewing sarcoma, as you can see. Very low mutation burden apart from EWS5. And different groups in the recent years have investigated in depth the genomics of Ewing sarcoma, mainly the group of Javed Khan, of Kim Stegmaier, and my group. And there are three main secondary mutations that can be observed: STAC2, I will come to this in a moment, P53, and CDKN2A. And the presence of those mutations is of adverse prognostic significance. There are also some copy number alteration. Most of them are linked to another translocation, which can be found in Ewing sarcoma between chromosome 1 and chromosome 16, And this 116 translocation is unbalanced and leads to gain of chromosome 1 and loss of chromosome 16q. And this is also of prognostic significance. Finally, the number of SNVs or SV is also of prognostic significance. And for SV, it could be related to the chromoplexy I told you about a minute ago, and that was described from Adam Schlien. In terms of intratumor heterogeneity, our work in the lab, as well as the work from other labs, from Stephen Lesnick and others, has shown that surprisingly, when you inhibit EWS5 by SI or SH RNA, you have a complete reversion. The cells stop growing but migrate a lot, as shown on the bottom in this video. So you see the two nodules on the bottom where cells migrate a lot and they are EWS5 low, whereas above just the nodules grow. And this led us a few years ago to investigate in depth these mechanisms and to think that might this be also observed in tumors where we could observe EWS5 high or low cells. And Georges Alain and Karine did fantastic work to come to this model where in Ewing sarcoma, there is a gradient of EWS5 activity, and some cells are EWS5 high or relatively high, and those cells proliferate. And you also have a minority of cells which have a lower level of expression of EWS5, and those cells are mostly mesenchymal-like, which is mesenchymal is a cell of origin of Ewing sarcoma, so they recover somehow their cell of origin phenotype, and those cells have cell matrix interaction and have a high propensity to migrate. This was further investigated in the lab by Marie Ming and Olivier using 10x genomic technology. And we could design a signature, which is called here IC-EWS, which is a signature of activity of EWS FLI. Mostly its activity on microsatellite sequences and target genes through microsatellite activation. And as you can see in the single cell, you have a different activity of EWS FLI. You have cells in red, which are high activity of EWS FLI. You have this loop, which are mostly cycling cells. And as you can see, they don't have the maximal activity of EWS FLI. And you have also cells in blue that have a low or a lower, because they still have activity of EWS FLI, a lower activity of EWS FLI. And as shown on the next slide, this is associated with some specific aspect of the cells. Cells with a high activity of EWS FLI are the OXPHOS metabolisms, where EWS FLI low have mostly a glucose catabolism with activation of IF1 type. And on the next slide, what we did here is to use different tumors, single cell analysis. Each dot here is a cells, and each color is a different tumors, and along the cells, depending upon the activity of EWS FLI along the x-axis, and plot the cell cycling activity. And as you can see on this, you have quite a range of activity of EWS FLI, but only a window of cells of activity of EWS FLI are cycling. Cells with a very high EWS FLI activity are not cycling, and cells with a low activity of EWS FLI are not cycling either. And on the next slide, this is true also for IF1 alpha, and those cells with this window, they have a low IF1 alpha transcription activity, whereas cells with a low EWS FLI or very high EWS FLI activity have a higher IF1 alpha activity. So this is also associated with sensitivity to treatment, and EWS FLI low have lower sensitivity to chemotherapy. So this is just something that we think is very important for the biology of Ewing sarcoma, is that EWS FLI activity can fluctuate in the cells. And this is a source of heterogeneity. The precise mechanisms of this is still under scrutiny, and it can affect either the transcript, the protein, or the activity of protein. It can be stochastic transcription. It has been shown by Heinrich, and we also showed this, that hypoxia influences stability of EWS FLI. As you can see, in the short term, hypoxia induce higher expression of EWS FLI, and in the long term, on the contrary, a lower expression of EWS FLI. And different groups, including those of Heinrich Cover and Bettloller and others, have shown also that this EWS FLI low activity can be due to antagonisms, particularly antagonisms through the EPO pathway or through the Wnt pathway. With this in mind, I will come to what I would like to present today, and work that was done by two fantastic guys in the lab, Thomas Grunwald and Didier Surdey. Both of them have now their own group, Thomas in Heidelberg and Didier in Zurich. And the question they were asking was, is EWS FLI acting alone in the Ewing cells? And Thomas investigated the role of germline genome architecture, and Didier mostly the role of secondary alteration, and in particular STAC2 mutation. So the starting point of the project of Thomas was that while Ewing sarcoma is not included in any predisposition syndrome, like Di-Fromeni and other, it has been known for decades that the epidemiological profile of Ewing sarcoma is very specific. And this was shown by the first one that showed that was Joe Fromeni in the early 70s, so 50 years ago. And the main conclusion that has been confirmed by many papers is that Ewing sarcoma is mostly observed in a population of European descent, and much rarer in African population, and kind of intermediary in Asian populations. So therefore, strongly suggesting that there is some kind of low penetrance genetic susceptibility to Ewing sarcoma. And this has been very nicely described on the next slide in a very recent paper by Logan Spector and Josh Schiffman, looking at the distribution of Ewing sarcoma in different countries. And as you can see, it's mostly in Europe. And what is also very interesting is that when you look at the USA, between white and black population, you have almost a 10 times difference in the frequency of Ewing sarcoma. And we investigated this aspect using a genome-wide association study on the next slide. So collecting through French and European collaboration a high number of germline DNA from Ewing samples, and then do the GWAS. And what is a GWAS? It is to look at hundreds of thousands of SNP across the genome, and to look at the frequency of those SNPs between Ewing patients and a population matched control. And to see, is there somewhere in the genome differences in the genome architecture between those two populations. And what is plotted here in the Manhattan plot is the inverse of the p-value. And as you can see, there are 6 regions where multiple SNPs point out the difference between patients and control. And Thomas addressed what's going on chromosome 10 here, and on this chromosome 10, I show you have a number of SNP that are all of them are in linkage disequilibrium and point out to this region. And Thomas could investigate different genes in there and pointed out to the EGR2 gene. So EGR2 is a transcription factor, also called CROX20, which has a role in the myelination process, as well as in the development of rhombomeres, and also some role in ossification and endochondral ossification. So first observation of Thomas was that indeed in Ewing sarcoma, EGR2 is highly expressed as compared to other pediatric cancers. This is clearly the tumor with the highest expression of EWS5. Second very important observation is that when EWS5 is knocked down in Ewing cells, there is a regression, decrease of the cell cycle, decrease of the ability to form foci. And even more importantly, on the right, what did Thomas to generate a Ewing cell line with an inducible small hairpin RNA against EGR2. And then when those cells were injected in mice, and after the beginning of the tumor, then the mice had some doxycycline in their drinking water. And as you see in the red curves, inhibition of EGR2 leads to complete regression of the tumor, so which was really very impressive. And third aspect is that EGR2 is a target of EWS5. So when we knock down EWS5 in Ewing cells, we decrease EGR2, and when we express EWS5 in heterologous system, we induce EGR2. So Thomas and Virginie, a research assistant in the lab, did a fantastic work sequencing the whole region in more than 200 patients, looking at the haplotype organization of all this, and looking at the epigenetic architecture. And in this region, you have 2 microsatellites named MSAT1 and MSAT2 here. MSAT1, we could not investigate this because it's a very complex structure, and we need some long-range sequencing to investigate this, which was not possible a few years ago. And Thomas focused on the second one, which is the MSAT2. And after a very nice analysis on the next slide, what it could show is that in a non-risk allele, so meaning that the allele which is present in controls, or which is more frequent in controls, you have this microsatellite, which is interrupted in red by a GGAT. And I told you that the more GTA you have, the more EWS is able to bind. And on the risk allele, on the contrary, this GGAT next is mutated or changed to a GGA. So therefore leading to connect the two GGA repeats, flanking the GGAT, and leading to very long GGAA stretch. And Thomas could show that there is a correlation between the genotype of the tumor and the expression level of EGR2. As shown, the tumors with AA genotype express more EGR2 than tumors with TT genotype or heterozygous. He could show that in reporter system, when he cloned the two alleles, the activation by EWS is much stronger with the GGAA than with the GGAT. And doing ChIP-seq analysis on the cell line, which is a Ewing cell line, which is heterozygous for the T and A allele, you could show that there is a strong binding of a much stronger binding of EWS on the A allele than on the T allele. So, which led to the conclusion that there is some kind of epistasis between EWS and some germline polymorphisms, and this led to the concept of transformation facilitating polymorphisms, meaning that this polymorphism by itself is not oncogenic, but it facilitates the transformation by EWS. And now with other colleagues and Mitch Macchiella at the NCI and Stephen Shannock and still Thomas Grunwald, we are investigating the other loci. And the data that we have show that at the other loci, also microsatellite sequences may play a considerable role to account for this susceptibility to Ewing. Now I will, and I believe that what we observe in Ewing sarcoma is probably something which is kind of more general in cancer. And that probably we do not at the present time take into account sufficiently the possible epistasis or interaction between the somatic alteration like EWS5 and the germline architecture of the patients. I will now shift gears and go to the second aspect that I would like to describe to you today, which is about the STAC2 mutation observed in Ewing sarcoma. So these are inactivating mutations, truncating mutation that completely knock out the genes. And this is of prognostic significance in Ewing sarcoma. Patients with STAC2 mutation fare less well than those without mutations. And there are some cases, and some of them were investigated with Armita Bahani at St. Jude. We could show that in some cases, tumors that are wild type at diagnosis get mutated at relapse, as shown here by the absence of the protein at relapse. So Didier worked with a very nice group of people, Sakina with a research assistant, and Sandrine and Veronique, who are fantastic computational biologists. And they generated knockout, this is a slide to indicate what is STAC2. So STAC2 is a member of the cohesin complex. Cohesin complex, I will come to the function in a moment. So it's a ring that can reel into DNA, which is formed by two main major proteins, structural protein SMC3 and SMC1A, that are connected to each other and that that have ATPase activity. RAD21 is very important, and they can contain either STAG1 or TAG2. And there are some ancillary proteins like NEPBL or WAPL, which are critical to load the coenzyme complex to DNA or to unload the coenzyme complex to DNA. Coenzyme has mostly two known roles. One, which led to the discovery of coenzyme initially, was its role in sister chromatid cohesion after replications. And with respect to this role, we initially thought that maybe a loss of function of STAG2 in Ewing sarcoma may lead to genetic instability. And in fact, when looking at SNVs and SV in Ewing sarcoma and comparing STAG2 wild type and STAG2 mutated cells, we could not observe any difference. So we thought that this was not how STAG2 was playing a role in Ewing sarcoma. The second role of STAG2 is to be able to form loops of DNA, which are very important for the tertiary, the 3D structure of the DNA. And there has been a lot of interest into this loop in the recent years, and it was suggested for long, but this has been demonstrated very recently by imaging technology that those loops arise through a loop extrusion mechanism, which is schematized here, where the loop, the ring, which is a coenzyme complex, can load onto the DNA, and the DNA progressively extrude through this ring until it reaches CTCF site, and then the loop is blocked when the ring, the coenzyme ring, encounter CTCF site. And with this function, we thought that loss of function of STAG2 obviously could lead to transcription dysregulation in Ewing sarcoma, adding to what EWS-FLI is doing. So Didier and colleague on the next slide generated a number of different models for STAG2, using CRISPR-Cas9 to inactivate STAG2 in wild-type STAG2 Ewing cell line, to rescue the mutation, which is indicated by the gray clone SA2R. And as you can see, the rescue, it's a good rescue, but the level of expression of STAG2 is a bit lower than what is observed in the initial STAG2 wild-type cells. And so in A673 cells, in TC71 cells, as well as knockdown experiments in 5 different cell lines. And using the stable clone, we could define a kind of core signature of STAG2 deletion of a bit more than 500 genes. And looking at the knockdown clone, we could show that this signature is shared by other cell lines, and even more importantly, that this is a signature that can be observed in the short term. As you can see, as soon as 24 hours after the knockdown, already this signature, either the downregulated genes or the upregulated genes, show up very nicely. And so with this core signature, we did some functional inference, looking at what could be the pathway dysregulated by STAG2 mutation. And very surprisingly, among a kind of agnostic stream of more than 10,000 different signatures, the one that showed up the most was this IC-EWS signature I told you about, which is the signature of activity of EWS-FLI, mostly through a microsatellite sequence. And among the 21st signature, 4 other signatures, which have been defined by colleagues in the field, also showed up very nicely. With the conclusion that one of the main activity of STAG2 deletion was to downregulate the activity of EWS-FLI, which was really very surprising. So we wanted to address the mechanisms of this. And obviously, the first hypothesis was, so this is just to show that in the short term, we already see very nicely the signature. First hypothesis was maybe a STAC2 mutation impact on EWS expression. And as you can see, this is clearly not the case in the different models. Does it impact on histone and histone active mark like K27 acetylated? And no. We then go a bit deeper on that and look at the binding of EWS, comparing STAC2 wild type and STAC2 mutated clones. And again, we only observe very subtle variation between the two, either at microsatellite or not non-microsatellite. Next, we investigated the super enhancer and K27 acetylated mark, and again, no real difference was observed between the two. And then we look at cohesin member binding to the DNA, and again, comparing for CTCF, RAT21, SMC1, and STAG1, we could not detect real difference apart from a small increase of the binding of STAG1, which is associated, if you look on the Western blots, with probably an increase of STAG1 when STAG2 is inactivated. So kind of compensatory increase of the expression of STAG1. And obviously for STAC2, we don't see anything in STAC2 mutated cells. So this was not a direct influence on either the expression level or the binding to DNA of those different guys. And so we decided to go to a kind of more in-depth analysis of the 3D structure of the genome. And for this, we used the IC technology. So with the IC technology, what is done is to cross-link a protein with DNA first, then to digest the DNA, and then to ligate the DNA fragment that are associated with the protein. Since IC is a global method of investigating the 3D structure of the genome, we used a more precise, maybe, method, which is CTCF I-chip. And in that case, what you do is to immunoprecipitate the protein of interest, and then look by deep sequencing at the DNA fragments that are linked to this. And we were interested to investigate what's going on at the level of the cohesin and the interaction between cohesin and CTCF. And so we did this I-chip analysis. So, and we look at this at different resolution. So at a low resolution, 250 kb resolution, so each dot is this size, we do not see any difference. So what you observe at this resolution is a compartmentalization of the genome between the A and B compartments, A being active euchromatin compartment, whereas B are heterochromatin inactive compartment. So no real difference at this level. We then investigated a bit deeper on this slide. So just to introduce what we observed with this. So we observed in red the region of interactions. So we observed the TAD, which are a region of higher interactions, which are shown here. We observed what is called here the corner peak, and the corner peak is the basis of the loop, where you have the interaction between two distant CTCF sites, the basis of the loop. And we were also quite surprised to see those stripes, which were described by IC. But here you can see those stripes. We see those stripes very, very nice. And to what correspond those stripes? So as you can see, maybe with those stripes, this is one CTCF site that can interact with the DNA until the next CTCF site. And the hypothesis for this, or for the stripe, is as you may know, the loop, at the basis of the loop, you have CTCF site on inverse orientation. And what we believe are those stripes is that the loop, when the loop forms, when it is attached initially to one of the CTCF sites. So as shown here, there is an anchor schematized where the loop begins at the left CTCF side and progressively extrude. And if you look at what would be the interaction in iChip, IC with this, you see the dot reds that progress along the X line with the progression of the loop. And you have to consider that when you do iChip, you look at a high number of cells, so a high number of loops at different stage. And when you pile up these signals, you have this stripe from one CTCF site in the good orientation to the next one. So what we're saying is that this stripe witness this anchored CTCF anchored loop extrusion process. And on the next slide, then we'll look at this is very nice slide that Didier designed, where you see the ring, you see the loop, and you will see with the progression of the loop, the progression of the signal on the iChip map. If you click, you will have an animation, and you see the progression of this. And so this is when the anchor is on the left CTCF site, and if it is on the right CTCF site, you will have a kind of Y band. You will have a Y band. So we speak about X stripe when this is the left CTCF, and Y stripe when the anchor is on the right CTCF. So usually the anchor can be indifferently in the left or right side. In the few loops where you have preference for either left or right, then on the anchor site you have a high number of cohesin, high decoration by cohesin. So what happens then between STAC2 wild type and STAC2 mutated cells? You see that when we look at the overall TAD structure, there's not much difference between wild type and STAC2 mutated cells. You can recognize the TAD in both. You can very well see the loop itself, the corner peak, well, the loop where you look at the basis of the loop. But what you observe is a strong decrease of those stripes. And this is rescued when the mutation is rescued. This is very different for STAG1 mutation. So we have STAG1 deletion mutant, so by CRISPR-Cas9. And as you see, you have a huge increase of the stripe in this condition, and this is also observed in the short term with siRNA experiment, where when you compare control and specific siRNA, you have a strong decrease of those stripes. Then the structure is conserved, but the stripes are decreased. This is true genome-wide, as you can see when we quantify the interaction at each loci. We see that in STAC2 mutant we have a decrease. When we rescue the mutation, we rescue partially this, probably linked because STAC2 is expressed a bit lower than in the wild type. When we mutate STAG1, we have a huge increase, and this is observed in two different cell lines. So the conclusion here was that STAC2 and STAC2 oppositely impact on the dynamics of the anchored loop extrusion process. But then the question of what is the relationship with EWS FLI? Well, how this may impact on EWS FLI? We hypothesize that in this process of anchored loop extrusion, what happens is that distant DNA elements are brought together in close vicinity during this process. And we hypothesized that maybe here, this is the way promoter-enhancer interaction can be enhanced and can be established. So to look at this, what we did is do the same analysis, iChip, with not CTCF this time, but H3K27 acetylated mark. So H3K27 acetylated mark both promoter and enhancer, and we thought that with iChip with this, we should be able to capture promoter and enhancer. And this was indeed the case. And when comparing wild type and STAC2 mutated cell lines, we observed a strong decrease of the interaction between promoter and enhancer. Here you have the promoter, which is marked by the H3K4 methyl-3 mark, and you have the different enhancer, promoter and enhancer, that are labeled with the H3K27 acetyl mark. And Véronique could design the promoter enhancer regulatory chain, so starting from the H3K4 methyl-3 mark, then looking at the interaction with the first enhancer, the more potent enhancer, and then from the enhancer 1 to enhancer 2, and enhancer 2 to enhancer 3, shown in this scheme above, and an example shown below with the DKK2 gene. And so you can design the regulatory transcription chain, and you can see which genes have this kind of microsatellite sequences, so EWS FLI1 bound sequences within the chain. And similarly to the stripe I showed you, we could show that there is a strong decrease. As you can see on the top, the H3K27 acetylated mark are not that different between wild type and STAC2 mutated, a very small decrease maybe in STAC2 mutated. But when you look at the interactions, you have a very strong, at least a 2 times decrease in the STAC2 mutated cells, in A673 and in TC71 cells, and you have a rescue with the rescue of the mutation. So the model at this point is that in STAC2 wild-type cells, you have these anchored extrusion mechanisms that can enable to establish promoter and enhancer interaction, as shown on the left. And if you lose this anchor, you lose this kind of scanning process, and you may have less ability to establish proper promoter and enhancer interaction. But the question is why? This is a genome-wide effect. So why does it impact on EWS FLI? And when looking at this interaction, what we noted is that these microsatellite sequences bound by EWS FLI have a higher number of interaction, promoter enhancer interaction, than either wild-type ETS sequences or any other promoter enhancer interactions throughout the genome. So that there is a particular high level of interaction at this level. And this was further shown on the next slide when we compared what happened with genes that are not regulated by EWS FLI, genes that are regulated by EWS FLI with a microsatellite bound site within the gene, and genes which are regulated by EWS FLI through binding at the promoter. And what you could see is that genes that are regulated by GGA within the genes are much more impacted by STAC2 deletion than other genes, and that genes that are regulated by EWS FLI at the promoter region, which therefore do not need any looping for EWS FLI to activate the promoter, are less impacted than the genes with a microsatellite within the gene. So I told you about this EWS FLI, low cells that have ability to migrate, and we thought that this is, let's look if those cells have this phenotype. And indeed, looking at the different cell lines, with one dealing experiment, we could show that indeed STAC2 mutated cells migrate much more than wild type cells. And this was also true for cell invasion. But then the question was, is this ability to migrate and to invade really related to the EWS low cells, or could that be a completely independent effect of STAC2? And for this, what we did is to take STAC2 knockout Ewing cell lines and overexpress EWS in those cells, with the idea that with overexpression of EWS5, we would be able to increase the level of activity of EWS5. And indeed, what is shown on the next slide is that we can rescue this migration effect when we overexpress EWS5. So altogether, we believe that this effect of STAC2 on loop exclusion, then on promoter enhancer interaction, and then on EWS activity is really dependent on EWS and this migratory effect, at least partly, but probably importantly dependent on this lower activity of EWS as a result of lowering of promoter and enhancer interaction. I'm coming to the end of my talk. So what is the clinical relevance of this? If we take those genes I told you about with a microsatellite within the chains, we observe that the downregulation of those genes in STAC2 mutant is of prognostic significance. What we believe is happening, I showed you this slide, in the context of a STAC2 mutant, what we believe is that the clouds of activity of EWS is shifted on the left, so that you still have a proportion of cells which are proliferating, and we have shown, as well as Kim Stegmaier, we have also been investigating this, that the proliferation of STAC2 mutated cells is not altered as compared to wild type. But in that case, you increase the number of cells with relatively EWS low signature. So just to wrap up a bit what I told you about, our current model for Ewing sarcoma development is that EWS is a major actor in this, on mesenchymal cells, and that in Ewing patients there are some transformation facilitating polymorphism that enable a kind of optimal activity of EWS. Then those cells are able to shift from EWS high to low. This shift is dependent upon mechanisms that need to be investigated in depth, and that STAC2 next is pushing a bit the system to the right, to EWS low, and this may account for metastasis and resistance to chemo. And this is interesting that Kim Stegmaier group could show that indeed STAC2 clones, STAC2 deficient clones, have an increased metastatic potential as compared to wild type. So some messages to conclude. The epistasis between germline and somatic mutations is probably something which is very important and somehow underestimated. Oncogenes can act qualitatively. They are frequently thought to work either on or off, but we believe that there is also a quantitative aspect that is very important for plasticity. STAC2 loss of function impact the extrusion mechanisms and the promoter interaction genome-wide, but particularly EWS, because this is a major transcription factor in those cells. And therefore, the role of STAC2 as in cancer, because STAC2 is mutated in different cancer, may be very highly context dependent, depending upon the key transcription factor that are expressed in the cells. And with this, I'd like to thank my colleagues, so obviously Thomas and Didier and colleagues, and also Frank Tirode, who did a lot on all of this when he was in the lab, and Franck has now his own group in Lyon also. Karine and Georges Alain, who have been very, very important on this collaboration with Mitch and Stephen at the NCI, which are very important. Obviously, collaborator on the GWAS and pathologist, clinician, patient, and their family. And also acknowledge Gilles Thomas, which was my mentor for my PhD, and I began my talk with this today, and who passed away a few years ago, and which was really a fantastic colleague. So thank you very much for your attention, and I'm happy to answer any question.
Thank you so much, Olivier. A really remarkable story, and fascinating to see the interplay of the germline and somatic genetic features. So again, just a reminder to everyone that we're at the top of the hour, but we will stay online for chat. So please feel free to submit as many questions as you like. We do have a couple that are related to the GWAS analysis. And first is from Wenan Chen. For the causal germline variant identified in the repetitive region after the GWAS, is the allele frequency higher in samples with European ancestry compared to samples with African ancestry? That is, could it explain the difference in ERG incidence between different populations?
So
sorry, EWS incidence.
Yeah, this is a very important question. For the one I described to you here, the EGR2, yes. But I would not say that this explained the difference between Caucasian and African in terms of risk to Ewing sarcoma. There might be also other factors. Obviously, as you may understand, it's not possible to compare directly the genome of European descent and African descent, because there would be many different polymorphisms and many confounding effects. But yes, for some of the polymorphisms, we observe a higher frequency in European than in African. But you also have to keep in mind here is that when you speak about African, there is a very high diversity of African, and it depends also on which population of African you're looking at. So I'm always a bit cautious when going from what we observe in the GWAS and what is the real cause of the highest frequency of Ewing sarcoma in European. But for this polymorphism, yes.
And you mentioned briefly the other loci that also appear to have very strong associations, very high p-values. Is there interaction between them? Is there a composite risk? Do people have more than one risk?
So yeah, this is something very important. We have looked a bit at this, but we believe that the series that we have, obviously Ewing sarcoma is a rare disease, and this is a GWAS only conducted with a few hundred samples, which is quite a lot for a rare disease, but which is nothing compared to the 10 of 1,000 or 100 of 1,000 of cases that are investigated in breast cancer. So at the present, we do not observe obvious interaction between the loci, but I don't think we have really the power to conclude on this.
So on the same theme, there's a question from Chiriclia Kalaidi. I hope I've pronounced your name okay. Could EGR2 be a target for treatment?
Well, this is an excellent point. So EGR2 by itself is a transcription factor. So as EWS fly, it's quite difficult to target it. But EGR2 is a transcription factor that receives the signals from, in particular, the FGF signaling. And so this is kind of one of the endpoints of such a signal. So this is something Thomas looked a bit on this, looking if inhibition of the FGFR pathway could impact, and it did. It can impact, but maybe not to the level we were hoping, if I may say. But yes, sure, this signaling pathway from the membrane to EGR2 is something important to consider, because EWS1 is activating EGR2, but also EGR2 has a kind of normal activation pathway. So there is a kind of cumulative activation of EGR2 through the transduction pathway and something additional by EWS1.
Okay. So another question for you, Olivia, if I can. The cohesin data are fascinating. Are you surprised that only STAG2 is altered and you don't see alterations or role of other cohesin complex members?
Clearly, we do not see other mutation in other cohesin complex. I think the overall biology of the complex and how this complex is loaded to the chromatin by an EPBL, how it is downloaded, if I may say, by WAPL, and the different stage of this, it's very complex. So our idea is that STAG2 plays a very specific role in this encore loop extrusion process. That clearly STAG1 has not the same role, and STAG1 mutation do not lead to the same alteration of the transcriptome, and so we believe that this is probably the key of the system, because it impacts not that much on the biology of the cells. If you inactivate CTCF, if you inactivate an EPBL or WAPL, probably you will get much stronger effect on the biology of the cell. So this is my yes.
Okay, well, thank you very much. That's the questions that we have. So thank you, everyone, for joining. And again, I encourage you to join again next week. And Olivia, thank you so much. Thanks for bearing with us with the little technical issue we had, but I think we got there in some style. So thanks again.
Thank you very much for the invitation.
It's been great. Hope to see you soon.
Yeah, bye-bye.